welcome back to Science Lectures 256. So if you didn't yet liked, subscribed and followed, please do so. Today we are going to look at the last part of our playlist, that is cell side, cell communication and cell division, that is cell division. So cell division is the process by which a cell divides into form daughter cells. The process by which a cell divides to form daughter cells. So there are two types of cell division. Number one is mitosis. Mitosis, and number two is meiosis. Meiosis. So mitosis is the process by which a cell divides to form two daughter cells. Then here meiosis, the cell divides to form four daughter cells. And mitosis happens in all body cells, what we call somatic cells. Then meiosis only happens in what we call the gonads. Somatic, all body cells except for reproductive cells. Reproductive cells are called gonads. So in mitosis, the genetic material is conserved. In meiosis, there is difference in genetic material. There is difference in genetic material. So in mitosis, the daughter cells are diploid, and meiosis, the daughter cells are haploid. Here we say the genetic material is changed by a half. Here the genetic material is maintained. And we discussed mitosis in our cell cycle. So today I want us to look at what they call meiosis. So we are going to look at meiosis. So meiosis happens in two stages. That is meiosis 1, then meiosis 2. But meiosis involves the 5 phases we saw: prophase, metaphase, anaphase, then telophase. But very important to note that meiosis happens in the gonads. Now let us look at meiosis 1. Meiosis 1 begins with prophase 1. So prophase 1 of meiosis 1 is analogous to the prophase we saw in mitosis, and we said that things like the chromatins now be condensed. They condense to form chromosomes. That is number 1. Then number 2, the nuclear membrane DC appears. We say the disappearing is a result of phosphorylation of the various nuclear proteins, things like lamins, H3A proteins that make up the nuclear membrane. They become phosphorylated, leading to its dissolution. Then this prophase 1, in fact, before I forget, it is the longest phase. Of meiosis. This is the longest phase. So in prophase 1, we have another special event called synapsis. So we said, if this is my cell, now the chromatins are condensed. They have condensed to form chromosomes. And now the nuclear membrane also appears. So what happens is this: if I'm to have a zoomed structure of this, that these chromosomes, this is a chromosome, shall pair up with another chromosome. Shall pair up with another chromosome. Let me use a different color. So if this is my chromosome A, it is going to pair up with chromosome B. Chromosome B. So this pairing up is what we call synapsis. So this pairing up is a process we call synapsis. And when they two pair up, they form what we call a tetrad. They form a tetrad. So when the two chromosomes form a tetrad. Tetra means 4. So this chromosome has 2 chromatids and this one has 2 chromatids. So they pair up to form a tetrad. Still, this prophase one has another event called crossing over. So during crossing over, we have exchange of homologues, or what they call different parts of a chromosome. So what do I mean? I'm assuming this is my one chromosome. So what happens is this, and it happened, these two chromosomes are joined at the point we call the chiasmata. So what happens is this. Part of this chromosome shall, use a different color, and also part of this chromosome here. So if this is another chromosome, part of it shall also be taken by this chromosome. This chromosome shall take this part and this chromosome shall take that part. And that is what we call crossing over. And these chromosomes, as we say, they are doing at a point called the chiasmata, over the chiasmata. And this is the major cause of genetic variation in organisms. The major cause of genetic variation in organisms. Exchange of parts of the chromosome during crossing over is a major contributor of genetic variation in organism, and it takes place during prophase 1 of meiosis 1. So after having looked at prophase 1, let us proceed to the next phase, and that is metaphase 1. Metaphase 1. So, as we saw in mitosis, we said in mitosis, the chromosomes align themselves at the metaphase plane. So, what happens is this. If this is my cell, remember there are things I've not talked about in prophase 1, but that do happen. Things like formation of spindle fibers, or what you call the microtubules. So these chromosomes, in fact, this tetrad shall align itself at the metaphase plate. We say the metaphase plate is just an imaginary line. So this is my tetrad. Remember, now we already have exchange of parts of homologous chromosome. Then we also have another one here. I didn't know that the science is clear. So this thing here contains 4 chromosomes. So instead of having chromatids align themselves at the metaphase plate, this time we have tetrads. Tetrads aligning themselves at the metaphase plate. When they align themselves, then the kinetochore. The kinetochore, we saw the ultrastructure of the chromosome. If you've not yet seen that, please go back and look out for the video. The kinetochore of each chromosome, Remember in mitosis we say that the kinetochore of each chromatid, but in this case it's the kinetochore of each chromosome, which is part of a tetrad. Is part of the tetrad attaches to the microtubules. So instead of having the chromosome, this time we have a tetrad with the kinetochore of each chromosome attaching the microtubules. So after that we proceed to anaphase 1. Anaphase 1. We said ana means a part. So anaphase 1. So this time instead of having chromatids moving to opposite ends of the cell. Instead of having chromatids moving to opposite ends, we shall have individual chromosomes. Moving to opposite ends of the cell. Moving to opposite ends of the cell. Shall have individual chromosomes moving to opposite ends of the cell. Then after that we shall have another process that is telophase. Another phase that is telophase 1. So during telophase 1, we say the cell will constrict. We shall have constriction of the cytoplasm. Constriction of the cytoplasm. Then we shall have the cell dividing to two daughter cells. We shall have the cell dividing to two daughter cells. By process, last time we said it's cytokinesis, but in meiosis we call it interkinesis. The cells divide into two daughter cells by process called interkinesis. So that is basically what happens in meiosis 1. We say during prophase we shall have things like synapses and crossing over. At metaphase we shall have tetrads aligning themselves at the metaphase plate instead of having individual chromosomes. During anaphase, chromosomes move to opposite ends of the cell instead of having individual chromatids. Then at telophase we shall have constriction of the cytoplasm and it dividing from two daughter cells. Then from there we proceed to meiosis 2. So meiosis 2 is not any different from what we saw in mitosis. Just that during meiosis 2, the cell does not repeat the process of interphase. So we discussed what is in interphase, G1, S, and G2. So if you've not yet seen that, please do so. Look out for that video. Then the cell begins directly from prophase 2. So in most of the cases, the nuclear membrane does not reappear. So we shall only have things like spindle fiber formation, that is spindle fiber formation, which spindle fiber formation is aided by the moving of centrioles to opposite ends of the cell. If this is my daughter cell, the centrioles shall move here, another move here, then they start producing spindle fibers. With spindle fibers, we say there are two types. They are also known as microtubules. There are two types: the polar microtubules and the astromicrotubules. So from here we proceed to metaphase. 2. Metaphase 2. So metaphase 2, just like we saw in mitosis, we have now our chromosomes, they align themselves. Our chromosomes shall align themselves at the metaphase plate. They will align themselves at the metaphase plate. Align themselves at the metaphase plate. Instead of having in meiosis 1 during metaphase 1, we show that we have tetrads. Here we have chromosomes aligning themselves at the metaphase plate. Aligning at the metaphase plate. Metaphase plate. Then from here we proceed to anaphase 2. So in anaphase 2, now we said we have what they call motor proteins. The motor proteins shall pull these chromatids. They shall pull these chromatids apart. The chromatids are part. They shall pull the chromatids apart. They shall pull the chromatids apart. Pull the chromatids apart. So when the chromatids are pulled apart, that means we can go to telophase 2. Telophase 2, where we shall have constriction again of the cytoplasm. Constriction of the cytoplasm. Constriction of the cytoplasm, that is number 1. On top of that, we shall have cytokinesis again happening. Cytokinesis happening, and at the end of the day, we shall end up with 4 daughter cells. 4 daughter cells. And each daughter cell is haploid or has half of the genetic material. So how do you restore half? When I say haploid, I mean N. So how do you restore the diploid number? Is when you shall have fusion of one haploid cell with another during a process called synthesis or fertilization, form 2N. I want us to look at lastly the uniqueness of meiosis. Like what makes meiosis different from other forms of cell division? What makes it different from mitosis? What makes it different from fission, as you saw in the earlier series? So number 1, meiosis happens in 2 stages. That is meiosis 1 and meiosis 2. That is number 1. Then we talk of the individual phases. That is prophase 1. In prophase 1, we have something unique which we call synapsis, where we have combination of 2 different chromosomes to form tetrads. Then number 2, we have what they call crossing over, where we have exchange of different parts of the chromosomes, forming new genetic combinations, bringing about genetic variation. Then we can also look at metaphase 1. Metaphase 1. So we said in mitosis, during metaphase 1, we have chromosomes. During metaphase, the chromosomes align themselves at the metaphase plate. Here we have tetrads. Aligning at the metaphase plate. Tetrads aligning at the metaphase plate. Then you can also talk of anaphase 1. So we say during anaphase, during mitosis, the chromatids are being pulled apart. In this case, we shall have chromosomes being pulled apart. And remember our order, we say we usually begin with chromatins. When the chromatins condense, they form chromosomes. So whenever they are being pulled apart, we shall form chromatids. So here, instead of having chromatids being pulled apart, we have chromosomes being pulled apart. Then still meiosis leads to formation of haploid cells. Haploid cells. While we say things like mitosis leads to formation of diploid cells. Then still meiosis leads to genetic variation. Meiosis leads to genetic variation. Meiosis leads to genetic variation. While in mitosis, as we saw earlier, there is no genetic variation because of the absence of synapses and crossing over, which leads to new genetic recombinations. So that is it for cell division. We thank you all for watching. And there is going to be a link in the description below. There is a video that makes the process of meiosis more visible with illustrations. So if you don't yet understand this, please hit the link and you see. Thank you very much.